Thanks committee for inviting me, and happy to share some work I done at the UCF. Currently, I'm a professor in the MSc department as well as the MPAC. I got my bachelor degree, master degree in physics, Ph.D. from biomedical engineering. I did my postdoc in the chemistry, come to UCF in 2003. My research has been focused on the soft materials. I'm particularly interested using the biological molecules as a building block to form an environmental response soft materials for biomedical applications. So today I'm going to share some work I done on the nanotubes, and as well as their applications in the sensors and as a drug delivery. Now, why I'm interested in the tubular structure? Tubular structure, as we know, often found in the nature and provide important biological functions. For example, lipid tubule, and which bridge the two cells, can be used as an information highway for the cell-cell communications. Microtubule and provide mechanical strength and for the cell. And virus nanotubule actually is an excellent gene delivery systems. Green bacterial antenna, which contain larger number of the nanotubules, which is can be used as a harvesting light and can convert light to electrical energy in the high efficiency. Those nanotubules found in the nature actually is based on the self-assembly of a biological molecule through the multiple non-covalent interactions. So inspired by those nature structures, and I had been working on using the synthetic molecules to design enzyme response nanotubes for the controlled drug delivery applications. And here is the lipid, which contain one chiral center and two hydrophobic tail. We found that this lipid actually can self-assemble into the vesicle, sulfuric vesicle. However, if we lower the temperature, the vesicle can convert to the helical tubules and eventually leads to the formation of the lipid tubule. The twist of the lipid tubules, twist of lipid sheets actually determined by the balance of elasticity and the chirality. Okay, the molecule is chiral. Chiral-chiral interaction is the driving force for bilayer sheets twist into the tubules. When the chiral interaction is able to overcome the penalty of the elasticity of the lipid bilayer, and tubule sheets actually twist into the tubular structure. The tubular structure actually is very good drug delivery systems, which contain, core, okay, and hydrophilic core and hydrophobic walls. So we have developed, colloid based on the tip, AFM tip, and can be used as a force probe or nano indent to study the mechanical properties of the lipid tube. We find the lipid tube is very rigid, so have the Young's modulate around 700 megapascal. Those lipid tube also can be uptaken by the cell and without cause cell death, or we can say highly biocompatible. And due to the hollow structure, lipid tube can encapsulate both hydrophilic and hydrophobic drug at the high encapsulation rate. Hydrophilic drug are expected load inside of the core of the tube, but hydrophobic drug will stay in the hydrophobic bilayer wall. The lipase, we know can be used to cleave the tail of the lipid. And what we find is the lipase actually can gradually degrade the lipid tube, start from the both end. Those the experimental result. So lipase actually is overexpressed on the disease side. So we expect those enzyme response lipid tube can be used to control the release of the drug on the demand. So lipid tube was injected the inflammatory side, and the overexpressed enzyme able to degrade the tube, trigger the release of the drug. Okay, so here is the cumulative release, show the drug with show without lipase release of the drug from the tube. In this case, drug only able to release from the both end slowly. However, in the presence of the lipase, drug release are significantly increased. Single injection, okay, here is the photographic image of a normal rods, foot of normal rods and arthritic rods. You can see the swelling. One single dose injection use the lipid tube with the hydrophilic drug, in this case located inside of lipid tube, can reduce the swelling. Lipid tube treatment use the lipid tube contain both drug can significantly reduce the swelling, okay, recover the damage of the tissue as well. So here's the summary or achievement. So we have developed lipase response lipid nanotube, which can encapsulate both hydrophobic and hydrophilic drug. The tubule can be uptaken by the cell without cause the cell death. The overexpressed enzyme in the disease side can be used as a trigger to control the release of the drug, treat the disease. And in the future, in the disease side, the enzyme variation is one factor. Also, pH will changes. And we're interested in develop the multiple response to drug delivery vehicle using the self-assembly of the polymer, okay, which we plan to synthesis. This polymer will respond to the pH as well as a response to the enzyme. So in this case, the drug relief vehicle and load by drug can be released from the drug delivery vehicle by using both pH variation and enzyme changes in the disease side. And I'm looking for the collaborations with the faculty which have experience or expert in the molecular dynamics simulation, and to understand the self-assembly mechanism of the biological molecules. I'm also looking for the collaboration with the faculty which have experience in the cancer design, so we can explore the application of the multiple drug response vehicle for treat the cancers. The second project I work on inspired by the green bacteria antenna, which I showed in the previous slide. And I'm interested in mimic this green bacteria antenna, using this, mimic the green bacteria antenna as a photo-induced electron transfer probe to detect the neuron transmitter. And we know the dopamine actually is a very important neuron transmitter. Concentration changes of the dopamine can cause the disease, such as Parkinson and Alzheimer's disease. So dopamine concentration level of the dopamine has commonly used as a biomarker to diagnose those diseases. And we use the biological molecule, okay, bioacid, have a member of the chiral center, which coupled with the cyanide and form a dimer. And this dimer can further self-assembly into a tube, nanotube, we call it J-aggregate nanotube. And due to the strong intermolecular coupling, or in this case, photo-induced electron transfer, can fast migrate through the tubules. And so due to the strong intermolecular coupling, you can see here from the adsorption spectra, due to the strong intermolecular interactions or coupling in the tube. And adsorption spectra intensify, shift to the right. Okay, shift suggests one very specific aggregation take place for the cyanide induced by the biological molecule. Here is the biacid, forms so-called J-aggregate, head to tail, head to tail, head to tail. So we use this very sharp intensity adsorption band of the nanotube as the optical probe to detect the dopamine. Okay, in this case, if we shine the light to the J-aggregate nanotube, photo-induced electron transfer can fastly migrate to the dopamine, leads to the drop of the adsorption intensity. So by plug intensity changes as a function of the dopamine, we find our detection limit can reach to the 0.4 nanomole, which is much lower compared to the electrochemical method and HPLC or, nanoparticle based on the optical probes. So this detection also can be visualized by naked eye. Okay, this is the tubule solution, appeared as the blue. If you add the dopamine, shine the light, photo-induced electron transfer take place, the solution become transparent. So can be easy to detect a dopamine. So another issue we addressed is selectivity. This is a J-aggregate nanotube based on the photoprobe. Not sensitive to the aspartic acid and the uric acid. Those are the two major interference species for detect dopamine because they are coexist with dopamine in vivo at much high concentration. So we find those sensors, those probe are not sensitive to the UA and AA. So to understand the detection mechanism, we calculated the HOMO and the LUMO level of the dopamine. And in the physiological pH, dopamine actually is oxidized. Oxidized dopamine can act as an electron acceptor. So to verify the photo-induced electron transfer do take place from the tube to the dopamine. So we calculated the HOMO and LUMO and HOMO level of a J-aggregate nanotube as well as the dopamine. You can see here the LUMO level of the dopamine actually it was in the gap between the LUMO and the HOMO level. So meet the requirement. Okay, so we find due to the strong interactions of a dye in the J-aggregate nanotube and photo-induced electron actually is delocalized, not confined individual molecules, and can lead to the fast photo-induced electron transfer. We find one dopamine can quench over 200 dyes inside of the nanotube. Okay, this is the mechanism for the high, leads to the high sensitivity, or we call the low detection limit. Here's the summary. Okay, so inspired by the green bacteria internal, we have developed the photo-responsive J-aggregate nanotube, which can be used as the photo-induced electron transfer probe for sensitive or selective detector dopamine. Okay, the future plan in the next 2 years. So, our hypothesis is, we know that amyloid beta peptide associated with the Alzheimer's disease and Parkinson's disease. The one hypothesis is amyloid beta peptide also can serve as a release agency for trigger the release of the dopamine from the neuron cell. So we are going to integrate the J-aggregate nanotube into the hydrogel to form a sensor papers and dip into the cell culture media. Use the amyloid beta peptide as a trigger to trigger the release of the dopamine from the cell. In this case, we can use this sensor paper inside to monitor the released kinetics of the dopamine from the cell triggered by the amyloid beta peptide. It is also my interest to see whether we can use this J-aggregate nanotube and to screen the drug. Okay, number of drugs have been developed for treat the Alzheimer's and Parkinson's disease and used as a releasing agency to trigger the release of the dopamine. So in this way, we can screen the drug very easily, use the J-aggregate nanotube integrate the hydrogel field. And due to the high efficient photo-induced electron transfer of a J-aggregate nanotube, they might also have an application in the photovoltaic device. And so I'm looking for the collaboration with the faculty which have expertise on the photovoltaic. I'm going to wrap up my presentation by acknowledging the student which made the contribution to this project, a tubule-based project. And a number of graduate students involved in these studies. So I collaborated with Dr. An in the MSc, working on understanding the mechanical properties of the lipid nanotube. I work with Dr. Guy from UPenn to understand the diffusion, how the hydrophilic drug diffused was inside of the lipid tube. For the detection of the dopamine from neuron cell, I'm currently working with Dr. Kai from Biomedical Science. I received the 3 NSF grants on this topic, which leads to the 37 journal publications. Happy to answer the questions you might have.